You can pass it around if you want to buy a candy bar. Whatever you want. All right, so with the cell cycle, focus please, everybody. So this is the general cell cycle, life cycle of a cell. Most of the time, your cell is in what stage of the cell cycle? Yeah, but what's all of this? Interphase, which is not part of mitosis, right? Interphase is what happens before and after mitosis. It's when a cell is doing what? What it's supposed to be doing, okay? Its job, it's working, it's doing its thing, it's doing whatever that kind of cell is supposed to be doing. Mitosis is really short. Remember that interphase has three parts. There's G1, and that's when the cell's just been born. So mostly what's it doing? It's growing a little bit, and it's doing its job, whatever its job is. And there's S, where the S stands for synthesis. And during S, what's it synthesizing? DNA. It's copying the DNA. It's when replication happens. It's still doing its job, though, whatever its job is. And then we get to G2, and it's almost ready to divide. Still working, though. What else needs to be copied? Organelles. So it makes extra copies of organelles. And then it's ready to go into what's called the M phase, which is mitosis, always followed by what? Cytokinesis. And remember that mitosis isn't really cell division. Mitosis is division of what? The DNA, the nucleus. And then cytokinesis actually splits up the cell. Okay? So both of those. Thank you. Got it. All right, so let's look at these questions here real quick. Mitosis is really a form of cloning, right? That's what mitosis does. Mitosis gives you exactly what you start with, two copies of exactly what you start with. So mitosis, if it's used for reproduction, it's what kind of reproduction? If mitosis is your method of reproduction, what kind of reproduction are you doing? Asexual, because you're getting what you started with. We know, though, that meiosis is more about our kind of reproduction, and it's sexual. And the biggest advantage of sexual reproduction is what? D, genetic variability, and the possibility that we can adapt and evolve and change. We know the biggest benefit of asexual reproduction is which one of those? It's fast. Doesn't require a whole lot of effort or energy. All right, just some terms to make sure you know. What's chromatin?
That's not unwound.
Yeah, DNA that's just sort of unwound and loose. And if it's unwound and loose, it's probably what? Working, being transcribed and translated. When it's not working is when it's all tied up in knots, right? When it's all tangled and coiled. What's a locus?
Location of location.
Yep, so location of a gene on a chromosome, where it's at. And somatic cells we know are just normal cells, right? Normal body cells. And at least in animals, are they diploid or haploid? Somatic cells. They're diploid. What's our symbol for diploid?
2N.
2N. 2 of each chromosome, right? 2 of each number. Germ cells we know are also diploid, but they're found where? At least in you. Where are your germ cells? In your ovaries if you're a woman, and in your testes if you're a guy. And they're the cells that know how to do what? What germ cells know how to do, but other cells don't. Meiosis. And in meiosis, one germ cell is going to divide how many times? It's going to divide twice to make how many cells? 4. And those 4 cells grow up to be gametes. And in terms of their chromosome number, they have how many chromosomes?
20.
Well, in us, 23, but like, what word am I looking for here? Haploid. Half the normal number. Half as many as normal. Two important terms on this slide to make sure you know. What are histones?
For the binding, like,
Yeah. Proteins that DNA wraps around. And they're found in eukaryotes. All eukaryotes have them. What other organisms have them besides eukaryotes? The archaea, it used to be called archaea bacteria, now they're just called archaea. So really the only groups that don't have histones are regular old bacteria, right? Eubacteria. They don't have them. Homologs or homologous chromosomes. We know those are chromosomes that come in pairs, right? You ladies have how many homologous pairs of chromosomes? 23 pairs. Us guys, on the other hand, have how many pairs? 22 pairs, and then 2 oddballs, right? An X and a Y that don't really go together at all. What's the advantage of having pairs and not singles? Yeah, a lot of times you can have a mutation and it not show up because a lot of mutations are recessive, right? And if you've got 2 copies and only one of them has a recessive mutation, you're still okay. That's why guys are more likely to get what kinds of things? X-linked diseases like color blindness and hemophilia, because we only got that 1 copy. And if it gets messed up, we're messed up. All right, what do we call that diagram that we're looking at there? It's a karyotype. And what we can't see is one of the pairs of chromosomes that's kind of important, and that's the 23rd pair. What's the 23rd pair usually? Those are the sex chromosomes. What are all the other pairs called? Autosomes. Because they don't have anything to do with sex. They're non-sex chromosomes, autosomes. We should have 2 of each. All right, so some of the roles of mitosis. Its job is to make new somatic cells. Those replace cells that have died or been damaged. They help you to grow. And in some things, maybe like a yeast, for example, that's how they reproduce. Remember that mitosis happens all over your body from head to toe. Meiosis, on the other hand, only happens in the gonads, the ovaries and the testes. It starts with a diploid germ cell and creates, by the time it's over, 4 haploid gametes. And then those go on to make babies. All right, let's talk about a little bit more about interphase here first. In interphase, we already talked about the 3 stages, G1, S, G2. It's really important that your body control the cell cycle and how it functions. Yeah, I should say no. But the idea here is if the cell cycle gets out of control, what happens? You get cancer or something bad. So you got to control it. And one of the ways the cell cycle is controlled is through the use of what are called checkpoints. And this is where a cell gets to a certain part of the cycle, and then it checks itself. And if everything's the way it's supposed to be, it proceeds. But if it's not, it either waits, or if things are really messed up, it does what? Apoptosis. It self-destructs. So the first checkpoint is around the end of G1, and if everything's right, it keeps on. If it goes past G1, that should say the point of no return, it'll self-destruct. And then there's another checkpoint in G2. And it's checking to make sure that by the end of G2, it's got 2 sets of DNA and 2 sets of what? Organelles. And if it does, then keep going. If it doesn't, wait or self-destruct. And then there's a third one that's right in the middle of mitosis rather. It's in metaphase. What should happen in metaphase? Where should the chromosomes be? In the middle, and they all ought to be attached up to what? Spindle fiber. If that is true, then it continues. If not, it either waits or self-destructs. So that's a way to sort of control the cell cycle through checkpoints. There's also some chemical signals involved in controlling the cell cycle. And we're going to simplify this diagram a little bit. What's the chemical we talked about that is like a signal for cell cycle? Cyclin. So cyclin is a signal. By itself, cyclin doesn't really do anything though. It's a signal that has to connect up to what to make something happen. It has to connect up to an enzyme called cyclin-dependent kinase, CDK. On their own, neither one of these are active. And when they get together, they become active. And what do we call them when they're together?
Maturation promotion factor.
MPF, maturation promoting factor. And that enzyme, that active enzyme, is a kinase. What do kinases do?
Phosphorylates.
They phosphorylate stuff. And in particular, this one phosphorylates what?
The nuclear.
Yeah, the framework that holds together the nuclear envelope. And when it gets phosphorylated several times, what happens to it? It breaks apart. And that's the beginning of prophase, right, when the nuclear envelope dissolves. So this helps to start mitosis, MPF. When it's done its job, what does MPF then do to itself? It breaks down the cyclin, so it starts over the process. So let's see if I have the diagram here I'm looking for. That's what I'm looking for. So early on in interphase, like G1 maybe, let's say this is G1 right here, G2, I'm sorry, G2S, G2, and so on. So there's a new baby cell has been formed right in there somewhere. There's almost no cyclin. But as the cell gets older, it makes more and more cyclin, and the levels go up and up and up and up and up and up. And then right here, right about the end of G2 or so, the cyclin gets together with CDK to make MPF, and it becomes active. This is when MPF is active. And it's phosphorylating nuclear membranes, making them dissolve, start prophase. And then when it's done its job, it destroys the cyclin, essentially resetting what? The biological clock. Because this is a new cell now, right? Because it's split. And it resets and starts over, and that pattern just continues over and over again. If your body was to make too much cyclin or too much CDK, that might cause what? Might cause cancer. Because your cells can divide too fast. So that's a cell signal mechanism for controlling the cell cycle. All right, so let's talk mitosis for a minute. The first actual stage of mitosis is which one? Prophase. Pro means first, right? So in prophase, we know that the chromosomes form, which means what lines up? The chromatin winds up around those histones, coils up. You can see the chromosomes. You've also got these organelles called centrosomes that make little spindle fibers that look like spider webs throughout your cell. The nuclear envelope starts to dissolve. The nucleolus disappears. And I'm grouping these two together because most textbooks still call this all the same thing, prophase and prometaphase. So by the time it's over, you can see chromosomes. The nuclear envelope is gone. There's spindle fibers formed in the cell, and the nucleolus is going away. That's prophase. Before we get there, though, here's a picture of a cell that's not dividing, which means it's in a what phase? Interphase. Not part of mitosis at all. And you can see it's got a distinct nuclear envelope. It's got a dark little nucleolus in the middle, and no spindles. That's an interphase cell. Here's the beginnings of prophase. You can see the nuclear envelope's gone. Chromosomes have wound up. The spindles are starting to form. So that's sort of at the beginning of prophase. Here's what it might look like at the end of prophase. So the spindles are all there. The chromosomes have attached up to them. What are the points on the chromosome called that connect up to the little spindles? It's part of the centromere. It's called the kinetochore. Why is it called kinetochore? What's kineto mean? Movement. So it moves and then it gets pulled, right? It gets pulled to where it's supposed to go. It's about moving the chromosomes. All right, and then after that we get metaphase. Meta means what? Middle. Chromosomes get pulled to the middle. Line up single file in the middle. Remember, this is where that third checkpoint happens. If they're all there and they're all lined up in the middle, keep going. If not, stop, wait, or self-destruct. So metaphase might look like that, except in your cells there would be a line of how many chromosomes? 46. And then after that we get what stage? Anaphase. Anaphase is always about what? Splitting. What's the more sciencey word? Segregation, right? Segregating, separating. And in this case, since it's mitosis, what gets segregated is just the two pieces of the bivalent chromosome, right? That breaks it in half. And one set of chromosomes starts getting pulled toward this side, one set toward the top. And what do we know about those two sets of chromosomes?
We do know stuff out here.
We do know some stuff about them. They're exactly the same. This is mitosis, right? Exactly the same. Cloning. All right. And then finally, we get to telophase, where those two sets made it to opposite sides. And after that, telophase is essentially the opposite of what phase? Prophase. Because we get a new envelope, a new nuclear envelope around each set of DNA. The little nucleolus comes back. The spindle fibers go away. Eventually the chromosomes are going to unwind, go back to being what? Chromatin. And already here in the middle, what's already started? But that's cytokinesis, right? The cleavage furrow is part of cytokinesis. It's got these little proteins that squeeze in on it, and eventually it's going to squeeze it into. Is that like what? It's the same kind of thing. You could call it invaginate, just means to pinch in, and that's pinching in, so you could call it invagination. But eventually it splits in two, and we get two cells that are exactly alike. And if we looked at if we could look at the chromosomes in there, what are the chromosomes going to be shaped like? Just on top of it, there, aren't you? Yeah, they're not going to be that anymore. Right? Mitosis' job is to split that in half. They're going to be more like that, right? Single-stranded, one molecule of DNA. Every form of cell division begins like that. Mitosis, meiosis, whatever, single-stranded chromosomes. So, during which stage of mitosis do the bivalent chromosomes separate into their constituent chromatids? I'll get you in a second. What's the answer here?
C.
Yeah, anaphase. Go ahead. Sure you can. All right, and the next one. During which stage of mitosis does the nuclear envelope disintegrate and the chromatin winds up to form chromosomes? That's prophase. A diploid cell with 36 chromosomes undergoes mitosis and cytokinesis. Mitosis and cytokinesis produce which of the following? They're going to make two cells, right? Mitosis just takes one and makes two. So it's either A or B. Which one is it? It's B. We know mitosis gives you the same number of chromosomes you started with. Started with 36, end with 36. And every kind of cell division should end with those one-stranded chromosomes, single-stranded chromosomes. So B's our best answer on this one. All right, and then this looks like the same question, except this one's about meiosis instead of mitosis. So what's our answer on this one? All right, we know that meiosis takes one cell and makes four. So it's going to put us at either C or D. We also know meiosis does what to the number of chromosomes? Halves it. We started with 36, we ought to get 18. And like we said a second ago, every form of cell division should end in one-stranded, one chromatid chromosomes. So D is the best answer on that one. 4 cells, half the number of chromosomes, single-stranded chromosomes. All right, and then this one. All of the cells of a multicellular plant have been observed to contain 27 chromosomes. This plant is composed of... The right answer, by the way, is not up there. Let me make a change. What do I need to change to make this be right? Yep. Well, C's already diploid, so let's do it here.
What did you say? They're hot. I mean, you could just took it off.
Nobody wants to see those legs. Put some clothes on.
Legs.
Put some clothes on.
I'll put you right now.
Anyway,
Throw down.
old man. Wow.
Let's take blood, spit on something.
Yeah. Mm-hmm.
For sure.
Mm-hmm. I'm gonna go Barney on you in a minute.
Oh,
I don't know what that means either, but anyway.
Fire grabbing heroin in his costume? Or?
What?
Yeah.
Did he really?
Yeah.
Yeah, you
Oh, well, didn't know that.
That's probably his use.
Anyway, the answer now is D. We know it's a plant.
He does heroin.
We know plants are eukaryotic because everything is eukaryotic except what? Everything is eukaryotic except bacteria. So it's got to be eukaryotic. And we know this is haploid because what? It's odd. Diploid means you got pairs, right? You can't have pairs if you got an odd number. So eukaryotic and haploid. You could have trisomy, but
Yeah.
you could.
I don't know.
You could.
You can switch heroin, you know.
All right.
That's heroin.
Shh, let's cut that. All right. So a couple of things here about cancer that we will mention quickly. We know that the cancer is when cells divide too fast. Normally, when two cells bump into each other, what does that signal them? Stop growing, stop dividing, you're on top of something else. But with cancer cells, that doesn't happen. They just keep on growing, keep on dividing, and they make this big clump, which we call what? A tumor. Now, one of the things that makes cancer bad is one, there's so many of those cells that they push other cells out of the way, they crowd them out. And the other thing really bad about it is that the cells are growing so fast that they never do what? They never actually mature. So they never actually start doing the job they were supposed to do in the first place. Because they never grow up, so they're useless, right? They never take on a role. Now, there's a word that you ought to, well, one thing you ought to know about cancer cells is they're sometimes considered to be immortal. They'll continue to grow forever as long as they're getting nutrients and food. Your normal cells are not. Because what's on the end of your little chromosome? Telomeres. And we know that every time your DNA replicates, what happens to the telomeres? They get a little shorter, and we think that's like your cell's built-in clock. And that eventually, when the telomeres get to a certain length, it tells the cell to what? Self-destruct. Die. Apoptosis. But the thing about cancer cells is they have this enzyme called telomerase. And what does telomerase do? It constantly makes the telomeres get what?
So does it make it
Longer.
or does it just like replace the parts?
It replaces the parts that get shorter. So as it gets shorter, then telomerase just takes it back to normal. So it's like they never age. And that's kind of scary. Like I said, there are cells from a woman named Henrietta Lacks that scientists are still to this day using to study tumors. She died in 1951, and they're still using her cells to study tumors.
So are those ones constantly growing even though she's dead?
Constantly, well, they're growing them on like petri dishes.
Yeah.
And yeah, they're constantly dividing, making new cells, and they will continue to do that forever as long as they have food and water and what they need because of this telomerase thing. Somebody say something? You hearing the voices again?
You're talking smack. The voices in your head are messing.
Yeah. All right, so a couple things about cancer. There's really two kinds of cancer. If you got to have cancer, the kind you want is benign. And what that means is that, yeah, you've got this tumor, but it's surrounded by this capsule almost. Which means, what about the cancerous cells? They can't really spread. They're just in that tumor. So the treatment for a benign tumor is typically what? Cut it out and you're okay, most of the time. On the other hand, you also have malignant. And these are the ones that can spread. And they can spread in lots of different ways. But the process in which they spread is called the metastasis. And one of the main ways they spread from one place to your body to another is through what set of tubes in your body? Not usually blood. The lymphatic tubes. The lymph vessels. And that's why if you have a tumor, one of the first things that they do when they're removing that tumor is they also look at what? They look at the lymph nodes near the tumor. And they're looking to see if those cancer cells have made it to those lymph nodes. Because if they have, that means they're already traveling, right? And they're already moving, and you may be in trouble. But if they're not in the lymph vessels, that means maybe it hasn't started spreading too much yet, and you might be okay. It might be one of those things where you cut it out, and you're going to be okay. Anyway, metastasis, this process in which cancer spreads from one place to another.
So is that why there's like different stages of cancers?
All right, we got a couple of minutes. We'll pull up meiosis here for just a minute. Not going to get too deep into meiosis today. Mention a little bit about it. We don't have one tomorrow morning. The other one this week is tomorrow afternoon.
Tomorrow, Thursday, Friday.
I think I originally didn't schedule them on those days because the band trip was going on. I thought we were going to be missing a lot of people. But we ended up not missing very many people.
Oh, well.
Anyway, let's look at this one slide for today. It's kind of the slide we stuck with a lot when we're talking about meiosis. We know meiosis has two main jobs. One of them is to take diploid germ cells and make haploid sex cells, right? Haploid gametes. And the other job is, while they're doing that, to create what? Genetic variability. All right. So, here's our diploid germ cell. It'd be in either the ovaries or the testes. Between the first picture and the second picture, the DNA got copied. So S phase of interphase happened. Replication. And notice the chromosomes went from being single-stranded to double-stranded. And what's the word for those double-stranded chromosomes? Bivalent. So it went from being monovalent up here to bivalent down here. Meiosis, remember, is a two. What's the word I'm looking for here? There's two main stages of meiosis. Meiosis 1, meiosis 2. In meiosis 1, it's mostly about separating what? The pairs. Remember that if this was one of your cells, this would say 2n equals what? 46. And meiosis 1's job is to get rid of the pairs. To take one member of each pair and put it into this cell, and take the other member of each pair and put it into that cell. So sometimes we say this is where segregation of homologous pairs happens. That's in meiosis 1. And in particular, the segregation part of that happens in which stage of meiosis 1? Which step of meiosis 1? Anaphase. Anaphase 1. Meiosis 2, on the other hand, happens to both of these cells. And it's meiosis 2's job to separate what? The pieces of the bivalent chromosome. It's going to split them in half. So each one of these cells down at the bottom, each one of these 4 cells at the bottom is going to get just one piece of each of these pairs. So that in you, this would say N equals what? 23. And each one of your gametes ends up with 23 single-stranded pieces of DNA. We will end today with this. We're going to talk just a minute about the very first stage of meiosis. And the very first stage is called prophase 1, right? It's the very first stage. Prophase 1 is really important because there's a couple of hugely important things that happen here. First of all, it's mostly like prophase in mitosis, though. What happens to the DNA?
Copy.
It's already copied. What happens to the DNA at this point?
It gets wrapped up.
It wraps up. It forms chromosomes. What happens to the nuclear envelope? Dissolves. What do the centrosomes do? They make spindles. Nucleolus goes away. All that normal prophase stuff happens. But in addition to the normal prophase stuff, this thing called synapsis happens. And in synapsis, what do we say synapse means over and over again?
Yeah. It's like
Connect. And in this case, what connects is each homologous chromosome finds its partner and they wrap together. That's called synapsis. And they'll stay wrapped for a while. What are the points that they actually connect together at called? Like right here and right here. Those are called kineto. That's not right. I just lost the word. Chiasmata. That's the word I'm looking for here.
You go.
Thank you. Chiasmata. And then they're going to unattach. But when they unattach, what sometimes happens? They break. And a piece of this blue chromosome ends up over here on this red one, and vice versa. And that's crossing over. That happens in prophase 1. And that is hugely important because where are those chromosomes going to end up that are crossing over? In the babies. And the parents end up giving chromosomes to their babies that they don't have. They give them combinations of chromosomes that they have, and that's where a whole lot of variation comes from, is crossing over. And that's in prophase number 1, and that's where we will pick up on this tomorrow.